And I would now like to invite Dr. Shook May Ho to the podium, who will lead us through session number 2.
Again, welcome and good morning. My name is Shook May Ho. University of Cincinnati has been a member for this committee for a while, and it truly is a great pleasure for me to introduce this wonderful session, which we're going to address like telomeres and telomeres, or any factors that is going to really be modulating all these telomeres that at the end of the chromosomes. So our first speaker is actually going to be a very exciting sort of lay down a foundation for us to address this question is Professor Maria Blasco. She's the director of the Spanish National Center for Research, and she's also the director of the Molecular Oncology Group, as well as the leader for the telomeres and telomerase research group. And the title of her talk is right on the board, and I would like to welcome her right now to the podium and give us a very exciting talk. I believe this is a teleconference. Yes,
hello,
welcome.
Hello, good morning, afternoon in Madrid. So for me, it's a pleasure to participate in this session. I hope you are hearing me there well. So I will be telling you about the role of telomeres and telomerase in cancer and aging. I'm sure you all know that telomeres are very important. This is the next slide. They are very important structures at the end of chromosomes. And next, please. We also know that telomeres, they shorten every time that a cell divides. Next slide. This generates, next please. So this generates, you have to go to the previous one, sorry. Okay, so this generates a problem at the chromosome end. So telomeres shortening with each cell division. This is cumulative with cell divisions, and this is known as the end replication problem. Of course, if cells would not have a way to compensate for this end replication problem, for instance, it would be impossible to maintain a species. So actually, next slide, there is an enzyme that is known as telomerase that is able to recognize chromosome ends, which you can see here in the drawing. And next, it can extend these chromosome ends de novo, therefore compensating for the end replication problem. We know now that telomerase is active through embryonic development, but after birth, telomerase is silenced, and this results next in the progressive shortening of telomeres with aging, and telomere shortening is considered one of the molecular causes of aging, because when telomeres get to a critically short length, this can induce at the cellular level cell death and senescence, and it can also result in organ dysfunction and aging. The exception to this are cancer cells. The next slide. Cancer cells, they have to activate telomerase in order to maintain their telomeres young and to be able to divide immortally. In the next slide, I show the same problem, but now we are looking at a telomere length versus the age of a donor, and we know that telomeres shorten associated to aging in every tissue that has been looked at, and this also includes the stem cells. And as I said, this telomere shortening is proposed to lead to the appearance of age-associated diseases. So there are a number of human diseases, next, that are characterized by, next slide, please. Okay, here. They are characterized by mutations in telomerase, and so due to these mutations in telomerase, these individuals, they are going to prematurely lose the generative capacity of tissues, and they also have a shorter lifespan. So this is proof that mutations in telomerase, so dysfunction in the capability of maintaining telomeres is going to lead to premature aging. We also know through the work of many different groups, Lisa Eppel, Blackburn, and others, that, next slide, telomere length could be also, sorry, can I have the previous one? Because I think, Okay, that telomere length can be also considered, or is being considered a possible marker for the rate of aging, and this is because it has been observed that individuals that are at the lower percentiles of telomere length, they have a higher risk of dying by cardiovascular disease, by infections, etc. Also, that there are many different environmental factors that can also influence the rate at which our telomeres shorten. In my laboratory, next slide, we have developed a technology which is a high-throughput technology that can be used in blood samples, which is able to very precisely quantify telomere length, but also allows the quantification of telomeres that are very short, what we call critically short telomeres. So this technology has been recently licensed to a company that is called LifeLength, and is being offering, next slide, this technology for diagnostics of these diseases, product development, or research. It also, there is an interest in trying to understand how telomere length per individual could be also important for personalized and preventive medicine. In this regard, companies such as LifeLength and others are offering telomere tests for individuals. Next slide. And the idea here is to compare telomere length of a given individual with that of a database of normal population to be able to see whether this individual has telomeres which are normal, or they are, let's say, shorter than normal or longer than normal. I'm just giving you an example in next slide of the kind of result that LifeLength provides, where you can see the black dot is an individual, and is plotted against the histogram of median telomere length as well as percentage of short telomeres. In this case, we are considering a short telomere those below 3 kb. And you can see that this particular individual is the normality. It's quite normal both for average telomere length and presence of short telomeres. Next slide. I think these tests have a potential use in personalized and preventive medicine. As you know, genetic testing can tell us the risk for a given disease, but they cannot tell us the effects of the environment and the interaction of the environment with these gene mutations for development of disease and time of onset of disease. And I think that telomere tests, next slide, can integrate these environmental effects, and the combination of both genetic tests and telomere tests could be useful in a more powerful prediction of risk and time of onset of diseases. And of course, next, this will also allow for development of new therapeutic strategies to prevent disease. So I'm going to give you a few examples of this. Next slide. In this case, is a familial breast cancer. This is a work published by one of the groups working here at the Spanish National Cancer Center. They are working in familial breast and ovarian cancer, and as you know, mutations in BRCA1 and BRCA2, they increase the susceptibility to this type of cancer. So what we have done is to combine the genetic information with the telomere length information, and it was seen that combining both information, this was a more powerful way to predict, in this case, the time of onset of disease. So those women that had the mutation and had shorter telomeres were developing breast cancer at earlier, they had an earlier onset of breast cancer and also ovarian cancer. Also following with these studies, next slide, we have also used quantification of telomere length and the presence of short telomeres on paraffin sections, and it's also, they are a good indicator. It correlates also with different parameters. For instance, hereditary tumors, they have a higher percentage of short telomeres than sporadic tumors, and this is particularly true in the case of the BRCX tumors, the ones that the gene mutated is still not known. And also they correlate the presence of these short telomeres. Next slide. It correlates with a number of histopathological findings, such as that the higher grade tumors have more of these short telomeres. In the case of the sporadic breast cancers, but not the hereditary breast cancers also having short telomeres, is typical of the estrogen receptor negative tumors. And in the case of the hereditary breast cancers, we also see a very significant correlation between expression of caspase-3, so the degree of apoptosis in the tumor, with the presence of these short telomeres. In the case of the BRCA tumors, next slide, we also see significant associations. Next slide, please. Okay, it's here. It's here. No, go back. Sorry, I don't see very well. Okay. We also see that there is significant associations between the presence of short telomeres and the expression of BCL2 or survivin. So I gave you these as examples of the possible use of this telomere test in the case of cancer. I wanted to give you another example, this one related with personalized medicine. Next slide, please. This is the case of the full sequencing of Dr. Michael Snyder, a researcher from Princeton. When he sequenced his full genome, he found out that he was mutant for telomerase, one of the mutations associated with aplastic anemia. He had looked at his telomeres by using a Sauter blot technique known as TRF, that is shown in the slide, and he could not see there any defects, any differences in telomere length with individuals of his same age. So he sent us his blot, and we could perform, next slide, our telomere test, looking both for average telomere length and the presence of critically short telomeres. In this case, are telomere spots that are shorter than 3 kb. And you can see here that he had quite normal average telomere length, is the black dot in the left. However, he was in the top percentiles of presence of short telomeres, indicating possibly as the result of the mutation in telomerase. Next slide, please. So I wanted also to discuss, of course, which is the evidence actually that telomere length determines disease and longevity, and of course the importance of the impact of life habits and lifestyle on telomere length. And for this, I wanted to highlight that it's very important, the concept of longitudinal studies. Next slide. The first, I think, longitudinal study over the whole lifespan of a species was published, I think it was early this year. In this case, was a study measuring telomere length in birds, in zebra finches, and the authors observed that telomere length at day 25 was actually predictive of longevity of these birds. So in my laboratory, we decided to perform a similar study, longitudinal study over the lifespan of a species. But in this case, we took as example the laboratory mice. Next slide. As you know, laboratory mice are used in biomedical studies to address the impact of different mutations in different diseases, and we thought it was interesting to see whether in mice could be also a good system to develop these longitudinal telomere length studies and to be able to study the impact of life habits and different treatments on mouse telomere length. So we apply our technology, the high-throughput QFISH, to individual mice. Next slide, we obtain blood from mice 4 times during the lifespan, and we follow the mice until the end of the lifespans. So the first thing that we use 2 different strains, a wild-type strain and a telomerase transgenic strain. And we observed that mice, their telomeres, even though they are much longer than human telomeres, as you can see in the bottom, they suffer telomere shortening with aging, which we can detect at 4-month intervals. And also with aging, you can see that in both strains, we can see the accumulation of these short telomeres. In the case of mouse telomeres, because they are longer, our threshold here is 15 kb, because they are much more heterogeneous and longer than human telomeres. Next slide, we could see that when we measure the rate of telomere shortening in mice, you have to remember that the rate of telomere shortening in humans is around 70 base pairs per year. Actually, we have performed a study in humans, and with this same technique, the rate was around 70 base pair per year, even though this may depend on the gender and other aspects. However, when we measured the rate of telomere shortening in mice, we found out that this was 100 times faster. So this was pointing us that even though mice have very long telomeres when they are born, they suffer a much faster rate of erosion of telomeres, and therefore this could be indicating that mice could also be a good model system to study the impact of life habits and environment on telomere length. So there we, next slide, set to address whether telomere length at any given time point in mice was also predictive of longevity. And contrary to the BERT study, we could not see that telomere length, both the average telomere length or the percentage of short telomeres, was predictive of mouse longevity at the different time points that we have studied. However, then we calculated the rate of telomere shortening, or the rate of increase of short telomeres with for individual mice in the next slide. And we found that in this case we could see a significant correlation between the rate of increase in the percentage of short telomeres and longevity in both strains, wild type and telomerase transgenic strain, but we could not see this correlation with the rate of mean telomere shortening per month. And this was because, I'm not showing the data, but this was because the rate of mean telomere shortening per month changes with the age of the mice. So it's much higher at younger ages than at older ages, and this produced that we do not see the correlation with the telomerase transgenic, since these mice, they have an increased health span, and there are less deaths during the early, the first year of the mice, and therefore this is why we not could see the correlation. But in the case of the increase in the percentage of short telomeres, we saw a significant correlation between lifespan and the increase in the percentage of short telomeres. So this suggests, next slide, that mice could be aging in part because they are also shortening their telomeres like humans. So the next part of my talk will be focused on whether we can actually delay aging in mice by changing telomerase activity. And in the past, and this is a work that was done when I was still in the laboratory of Carol Grader, we generated a telomerase knockout, mice deficient for telomerase, and in this artificial model, we saw that telomere dysfunction produced by telomerase deficiency was sufficient to produce aging in every tissue of the mice that has been studied. And also it served as proof of concept that telomerase could be a good anti-cancer target, because these mice also, because they had less telomerase, they had less cancer. I also generated, next slide, a transgenic, which I already mentioned before, a mouse model transgenic for telomerase, in this case in different epithelial tissues in the mouse. And we found that increasing telomerase in adult tissues resulted in a better tissue fitness, but also resulted in a slightly higher incidence of cancer. So this is summarized in the next slide. You can see that if you artificially eliminate telomerase from mice, these mice are going to develop premature aging and are going to be more resistant to cancer. And if you increase telomerase, there is less aging apparently, but because there is more cancer, we could not see any impact on lifespan in these mice. So a few years ago, we decided to combine telomerase transgenic expression with increased expression of tumor suppressor genes, such as P53, P16, and P19R, and this was in collaboration with a group of Manuel Serrano, also here at the CNIO. These mice have 4 different transgenes, and just to summarize, I will call them super mice because they have been, but they have 4 different transgenes. The idea was that if we would be able to increase telomerase in a cancer-resistant background, we would be. Next slide, please. Oh, I think you are not seeing. Okay, next. we will be able to extend longevity. And this was the case. As I said, this is already an old study, but you can see that by combining increased telomerase activity with increased tumor suppression, we were able to extend the median lifespan of mice by 40%. And you can see in the bottom that the percentage of mice alive at 3 years of age was very highly increased in the case of the mice with increased telomerase and increased cancer suppression compared to the control mice. Next slide. We were not only increasing median longevity, but if we look at the time of life of the mice without any death, which I'm considering here health span, we were able next to triplicate this time of life of mice free of disease. So increasing telomerase and tumor suppression was increasing what is known as the health span. Next slide are some examples of tests that we performed to prove that these mice have an increased health span. This is a neuromuscular coordination test. Here we measure the ability of mice to hold onto the bar, and you can see that this is something that chronologically young mice do very well, but as mice age, they start falling from the bar and failing the test. However, the mice that were transgenic, even though they were chronologically old, they were able to pass this test the same success rate than the young mice. Also, next, they had a better glucose tolerance compared to the control mice. Next slide. Okay, we also looked at the aging in the skin, in this case by looking at the subcutaneous fat layer, which is a biomarker also of aging in mice. And we saw that in control mice, the mice that were chronologically old, they had a thinner subcutaneous fat layer compared to the wild-type mice. However, this thinning of the subcutaneous fat layer was not seen in the mice that were transgenic for telomerase and the tumor suppressors, also indicating that these mice, even though they were the same age, they had an increased fitness or health span in the skin. And this can be seen, next slide, by simply looking at the mice. In this case, you can see two mice of the same age. The one in the left is a control mouse. You can see graying of the hair and alopecia. And the one in the right is the telomerase transgenic with increased tumor suppression, which clearly showed less skin aging. So, of course, this is indicating that increasing telomerase at the same time that we increase cancer suppression can significantly impact on the rate of aging by increasing health span and also increasing median longevity. However, the interesting thing here would be, next slide, to be able to envision a therapeutic strategy where we can treat mice with telomerase so that we can delay aging but without increasing cancer at the same time. And for this, next slide, we developed the idea that aging was in part produced in mice because they lack telomerase in adult tissues, the same as humans. And this telomerase deficiency was causative of aging in mice, telomere shortening and aging in mice. Therefore, if we will be able to reactivate telomerase in mice when they are middle-aged or they are old, we will be able to increase health span and delay aging without increasing cancer. And for this, we develop a gene therapy by using adeno-associated viruses. Next slide. As you may know, adeno-associated viruses are being used right now in several clinical trials, and actually there is one already that has been approved for treatment. And they are the state-of-the-art gene therapy nowadays because the adeno-associated viruses, they are non-integrative and they are very little immunogenic, and they are considered quite safe. So our idea was to use a vector. Next slide. A couple of vectors. One was the telomerase vector, and the other one a GFP vector, to follow the expression of the within the cells that were transduced by the vector. And you can see here that when we treat the mice with the EGFP gene therapy, this particular serotype, AAV9, it infects many different mouse tissues, and we decided to use this serotype because we wanted to see broad effects of telomerase re-expression in health span and longevity in mice. Next slide, you can see the percentage of transduction in different, next slide, in different mouse tissues, which was similar to that has been described for this particular type of vectors, the AAV9. So the experiment actually is in the next slide. We decided to treat two groups of mice, one that was middle age, 1 year of age, the other one that was 2 years of age, with a single treatment with either the GFP or the telomerase vector. We were able to see that telomerase RNA, telomerase protein, was increased in different tissues of the mouse after treatment. Next slide. Telomerase activity, in this case we are looking at telomerase activity in the lung, was also reactivated in vivo. In this case you can see that the mice treated with the GFP vectors, they do not have telomerase activity in the lung when they are old, 2 year old. However, the mice treated with telomerase, with a single treatment, we were able to reactivate telomerase in the lung. And so this per se is already of interest because one of the diseases associated to mutations in telomerase, next, is pulmonary fibrosis. So this could be a possible therapeutic strategy also to explore in the case of these diseases produced by mutations in telomerase. Next slide. So we perform again a number of tests to see the health status of these mice that have been treated with the control vector or the telomerase vector, and we observe beneficial effects of treating with telomerase in the case of bone density, the thickness of the subcutaneous fat layer. Next slide. Also in the neuromuscular coordination assay, we saw an impact of the telomerase treatment in the case of the mice at 12 months, more or less, after the treatment. When the control mice already show a decline in this test, the ones that were treated with telomerase, they maintained their ability to perform this test. We also saw, next slide, the beneficial effect in rotarod tests in the mice that were of the 2 year old group. And probably more important, in the next slide, as an indication of the beneficial effects of telomerase treatment, we saw an increase in the median and maximum lifespan in both age groups, in the 1 year old and the 2 year old group. And this is already indicating that in this setting, telomerase doesn't seem to be increasing cancer, because actually these mice have an increased longevity. And in fact, next slide, when we measure the incidence of different malignant tumors in mice, we see that the mice treated with telomerase did not have an increased cancer incidence compared to the control mice. So we were interested in seeing which is the pathway by which, next slide, telomerase is delaying aging in mice. And of course it can be through its ability to elongate short telomeres. This is the canonical role of telomerase, extending short telomeres. And therefore this could be then delaying the onset of aging, but it could also be next to the known role also of telomerase, the proposed role of telomerase as an activator of Wnt pathway target genes. And therefore we explore both possibilities to explore for short telomeres. Next slide, we look at telomere length in different tissues, in the control group or the telomerase treated group, and we observed that in all different tissues that we have looked at, we saw a decrease in the percentage of short telomeres in the mice treated with telomerase. So this would indicate that telomerase reactivation is indeed preventing the accumulation of short telomeres. Next slide. We also saw, however, that as expected, telomerase overexpression was also activating beta-catenin, as it has been described, in different tissues, and also some of the target genes, which I'm not showing here, such as cyclin D1. So next slide, in order to distinguish between these two pathways, what we did is to use now a treatment in which we treated the mice with a dominant negative mutant of telomerase, which is catalytically inactive, so it's not able to extend short telomeres, but it still has been shown to be able to activate the beta-catenin pathway. Next slide. This is the slide shows the mutation in the catalytic site of telomerase, and also the fact that this dominant negative is also able to upregulate cyclin D1, even though it's catalytically dead for telomerase activity. Next, you can see that this mutant, however, is not able to rescue short telomeres in mice in vivo, because we see that while the percentage of short telomeres is rescued in mice treated with telomerase wild type, is not rescued in mice treated with the telomerase dominant negative. Next slide. Similar, the aging-associated pathologies like such as osteoporosis or in this case insulin resistance, we see that only the mice treated with the wild-type telomerase, but not the mice treated with the dominant negative telomerase, show an improvement in these parameters, suggesting that only wild-type telomerase was able to have this effect in delaying aging or extending health span. Next, when we look at longevity, it was very clear that only the wild-type telomerase treatment was able to extend longevity, but not the dominant negative telomerase treatment. So we think that these results, next slide, indicate that telomerase gene therapy is delaying aging and increasing health span through the ability of telomerase to recognize short telomeres and elongate short telomeres, and in this manner is preventing or delaying the occurrence of age-associated pathologies. Next slide. So I think this can be put in the context of trying to understand really which is the molecular cause of aging in order to treat more efficiently or to prevent all the diseases that are associated with aging, and this includes cancer, cardiovascular, diabetes. I think this proves that a single intervention, a single gene intervention, can have systemic effects because it's targeting one of the molecular causes of aging. Next. So that telomerase would be, as it has been described for other pathways, could be an anti-aging intervention, and by extending the percentage of short, by extending telomeres and preventing the accumulation of this particular type of DNA damage, is also having an impact, a global impact in the process of aging. And I will finish here with the people of the lab. Next slide, please. Next slide. Thank you. So these are the people of my laboratory in Madrid, and I particularly want to thank Bruno Bernardes, who is the one that did the studies on the telomerase gene therapy, as well as longitudinal mouse studies. Thank you very much.
Thank you, Maria. That was a fabulous talk. Can I ask a question? Will you be able to stay for the discussion later on at the end of the session that is around, that's going to start around 12:10?
Unfortunately,
will you be able to?
I will have to leave, unfortunately.
Okay. So in that case, why don't we have two questions here and see whether we can have two questions. I saw. Can you identify yourself, please?
John Moran, University of Michigan. That was a very nice talk, and I like your super mouse. My question is, have you tried the reciprocal experiment? Have you overexpressed telomerase in a sensitized cancer background? And then do you actually get more tumors? Because that could have implications for the latter studies you were talking about.
That's a great question. We are doing that. We are doing that precisely right now. We're using this gene therapy strategy to address whether, in the setting of a tumor model, such as, for instance, KRAS-induced lung cancer, telomerase is promoting or delaying lung cancer. I think this question has not been clearly answered yet, and we are doing that right now.
Thank you.
I'm Larry Baker, also from the University of Michigan. I agree with John. It was very exciting to listen to you. I'd like to ask you, though, specifically about the cancers that were induced in the mice. There are, for example, in men, as they get older, 70% of men in the 70s get prostate cancer, 80% of men in the 80s get prostate cancer. But that is not a clinically dangerous disease most of the time. So is the cancer that's occurring in these mice, is that a cancer that's going to shorten their life?
Yeah, I think wild-type laboratory mice, they usually develop different cancers than humans. They are more similar to pediatric cancer, so usually mice die from sarcoma or lymphoma, and it's very rare to find other types of cancers in mice unless you have some particular genetic alteration that could be favoring the appearance of epithelial type tumors.
But you also
I don't know if I answered your question.
You did, but you also listed adenoma, and that's not a cancer.
Yes, it's true. There was, in one of the mice, there was, but I think it probably was a mistake. I think it was an adenocarcinoma. I think it was a lung adenocarcinoma, but I don't know if I can go back in my slides or not. Probably not.
Okay, Maria, I really want to thank you, but before you leave, I'm a very selfish person. I saved myself 1 minute. So to ask you a very important question, and that is related to the whole field of these induced pluripotent stem cells. So when you do all these experiments with these induced pluripotent stem cells, how would you perceive that the telomeres are going to work and telomere is going to work when you put those cells back as either therapy or tissue regeneration?
Yeah, my lab has contributed with several works to this field of induced pluripotent stem cells and telomeres. So we actually show that telomeres were elongated. This was also shown by others, but we show that telomerase activity was necessary for this telomere elongation with reprogramming. And also recently we are very much using this reprogramming system to try to identify factors that are important for regulating telomere length in vivo. And we identify and published a couple of years ago that one of the telomere binding protein that is known as TPP1 is also essential for telomere elongation in vivo. So a cell mutant in TPP1, it can be reprogrammed, but these reprogrammed cells will not elongate telomeres. So it's like lacking telomerase. So this is a great system for us to try to find factors that are important for telomere length regulation.
So is it safe to say that when we put back the induced pluripotent cells, the iPS cells, into a host, then they will actually be in the rejuvenile state in terms of telomere?
Yes, what we describe, and this was a paper in Cell Stem Cell, if you want to check it, in 2009, we described that at the time of isolation of the clones, of the iPS clones, we saw a very clear telomere elongation, but it was not, it did not reach the hyperlong telomeres of ES cells, isolated from a blastocyst. And in fact, we have shown that is post-reprogramming and associated with cell passaging, where iPS cells acquire these hyperlong telomeres that ES cells have. So somehow, very long telomeres characteristic of pluripotent mouse stem cells. This was done with mouse cells. In part, are acquired post-reprogramming and associated to cell division.
Thank you very much. I think we have to end this right now. We can, okay, so we have a little bit of program change. Thank you so much. We're going to have a break right now.
Pleasure.
Thank you. Thank you for, 10 minutes break, please. That's perfect.